Hey there, curious minds. Welcome to the Deep Dive, where we sift through the sources you've shared and pull out those essential nuggets of knowledge, giving you that aha moment without the information overload.
Great to be here.
Today we're taking a deep dive into the foundational world of plant genetics, drawing directly from the pages of Raven Biology of Plants.
It's a truly rich and honestly captivating topic. Our mission today is to give you a clear, accessible summary of how characteristics get passed down through generations in eukaryotes, organisms with complex cells, especially plants.
Right.
We'll explore the intricate mechanisms behind both sexual and asexual reproduction. Everything from the microscopic world of chromosomes to Mendel's groundbreaking discoveries, and even how tiny changes drive evolution.
Exactly. Think of this as your essential guide to understanding heredity in the plant kingdom, explained in plain language. We'll walk you through each major concept, finding, example, all the crucial details from the text.
We'll try to make it feel less like a textbook reading and more like a chat about some amazing biology.
Absolutely. So prepare to see how plants manage these impressive biological feats.
Okay, let's unpack this. Let's kick things off with a fundamental question. Have you ever wondered why dandelions always produce more dandelions and oak trees always grow from oak acorns?
It seems obvious, right?
Yeah, but that age-old observation about offspring resembling their parents, that's heredity. And the scientific study of it, genetics, only really got going in the second half of the 19th century. Relatively recent when you think about it.
That's a great starting point. And while early genetics looked at lots of organisms, our focus today is specifically on eukaryotes, those organisms whose cells have a nucleus, and mostly on plants. We're delving into what's often called Mendelian genetics.
Named after Gregor Mendel, who we'll definitely get to.
Oh, absolutely. His work was foundational.
And at the very core of eukaryogenetics, especially in plants, is sexual reproduction. It's kind of a defining feature, isn't it?
It really is. It's a complex process, maybe more complex than asexual, but it's absolutely central to the diversity of life we see.
So it's not just one event. No, not at all. What's fascinating here is that sexual reproduction isn't a single thing, but an elegant cycle. It involves two critical processes constantly alternating: meiosis and fertilization.
Okay, meiosis. Break that down for us.
Right. Meiosis is a special kind of cell division. Its main job is to have the chromosome number. Imagine a cell having two full sets of genetic instructions. We call that diploid, or 2N.
Two sets, got it.
Meiosis takes that diploid cell and, through 2 rounds of division, produces 4 daughter cells. But here's the key. Each of these daughter cells has only one set of instructions. They're haploid, or N.
So if diploid is like having 2 copies of a whole recipe book, meiosis makes 4 recipe cards, each with only half the instructions.
That's a great analogy. And in plants, these haploid cells are often spores specifically. Meiospores produced in structures like flowers or cones. Then, to complete the cycle, you need fertilization.
Right, the other half of the cycle.
Precisely. Fertilization, sometimes called syngamy, is when 2 of those haploid recipe cards, which we call gametes, fuse together.
Male and female gametes, usually.
Exactly. This fusion creates a brand new diploid cell called a zygote, restoring the full 2 sets of genetic instructions. This whole dance ensures that the chromosome number stays constant generation after generation. It's really quite elegant.
It is, and crucial to this whole system are homologous chromosomes, right? Or homologs.
Yes, homologs. If you could peer inside a diploid cell, you'd see its chromosomes aren't just jumbled, they're arranged in pairs. One chromosome in each pair originally came from one parent's gamete, the other from the other parent's gamete.
Like matching socks.
Yeah, kind of like matching socks. They're similar in size, shape, and they carry genes for the same types of traits, like flower color or seed shape, although the specific versions of those genes, the alleles, might differ.
Okay, that makes sense. Now, you mentioned chromosomes. These structures themselves are pretty amazing feats of packaging, aren't they?
Oh, absolutely. The eukaryotic chromosome is an incredible feat of natural engineering, especially how DNA is packed. Each chromosome is essentially one really, really long, continuous thread of double-stranded DNA tightly wound up with proteins.
How long are we talking?
Get this. A single diploid cell from a wake robin plant Trillium can contain about 68 meters of DNA.
68 meters. In one microscopic cell. How?
Exactly. To fit that much material into such a tiny space, you need incredibly efficient packing. The DNA doesn't just float freely. It combines with special proteins to form chromatin.
Chromatin. That's the stuff that looks like colored threads under a microscope.
That's the one, yeah. It stains well, hence the name. More than half of chromatin is actually protein, and the most important ones are these small, positively charged proteins called histones. 5 main types.
Positively charged, and DNA is negatively charged, right? So they attract.
Precisely. Think of histones as tiny spools. The negatively charged DNA wraps around them like thread around a spool. These basic packing units are called nucleosomes.
Nucleosomes.
If you could see them, they'd look a bit like beads on a string. Each bead is a core of 8 histone molecules, 2 each of 4 types with the DNA filament wrapped around it almost twice. Then a 5th type of histone, H1, kind of clamps onto the outside.
And this wrapping shortens the DNA.
Significantly.
Yeah.
By about 1/6, just at this first level.
But 68 meters down to 1/6 is still a lot.
It is, so the packing doesn't stop there. These nucleosomes then coil up further, forming a thicker 30-nanometer fiber. Then that fiber forms loops, which condense even more. It's a multi-stage process. Eventually, you get the super compact metaphase chromosomes that are visible during cell division. It's just incredible engineering.
Wow. Okay, so DNA's packed. Now let's get back to that special division. Meiosis. The dance, as you called it.
Right, the dance of meiosis. It's absolutely vital for creating genetic diversity through sexual reproduction. And it doesn't happen in just any cell. It's reserved for specialized diploid cells and only at particular times in an organism's life cycle.
And the outcome.
The outcome, as we said, is 4 haploid cells from one diploid cell. These haploid cells might become gametes, destined to fuse with another gamete. Or in many plants and fungi, they become myospores, which can develop into a new organism or part of one, all by themselves without fusing.
Interesting. And meiosis happens in 2 big stages, meiosis first and meiosis the second.
Correct. 2 successive nuclear divisions. Meiosis first is really the unique one. Its main job is separating those homologous chromosome pairs.
Okay, walk us through meiosis the third.
It starts with prophase 1. This is often a very long and complex phase. First, the chromosomes condense and become visible like long threads. But remember, the DNA already replicated before meiosis started, so each chromosome actually consists of 2 identical sister chromatids joined at a point called the centromere. They just look like single threads initially.
Okay, duplicated chromosomes, then what?
Then comes something truly unique to meiosis. Synapsis. Those homologous chromosomes, the matching pair from each parent, find each other and pair up precisely gene by gene along their entire length. It's like they're zippering together.
Wow, that sounds crucial.
It absolutely is. These paired homologous chromosomes are now called bivalents. And at this stage, each bivalent actually consists of 4 chromatids all associated together. A protein structure called the synaptonemal complex forms between them, holding them tightly aligned.
Okay, paired up tightly. Is that when the shuffling happens?
Exactly. This is where crossing over occurs. While the homologs are paired in synapsis, portions of chromatids can actually break and exchange segments with the corresponding chromatid from the homologous chromosome. Importantly, it's between non-sister chromatids, one from mom's homolog, one from dad's.
So bits of mom's chromosome get swapped onto dad's and vice versa.
Precisely. This physically shuffles genetic material, creating new combinations of alleles on a single chromosome. It's a major source of genetic variation. You can often see the physical evidence of these crossovers later in prophase I as X-shaped structures called chiasmata. These chiasmata also help hold the homologs together after the synaptonemal complex breaks down. Then the nuclear envelope breaks down, preparing for the next step.
Which is metaphase I.
Correct. In metaphase I, the spindle fibers' microtubule tracts form and attach to the centromeres of the chromosomes within each bivalent. These paired homologous chromosomes then move to the center of the cell and line up on the equatorial plane.
How is this different from regular cell division, mitosis?
Key difference. In metaphase I of meiosis, the bivalents, the pairs of homologs, line up. The centromere of one homolog lies on one side of the equator, and the centromere of its partner lies on the other side. In mitosis, individual chromosomes line up with their centromeres directly on the equatorial plane.
Ah, okay. Pairs line up across the middle in meiosis the sit, then anaphase the fifth.
In anaphase Y, the homologous chromosomes within each pair separate and are pulled to opposite poles of the cell by the spindle fibers. But, and this is critical, the centromeres do not split. The sister chromatids remain attached to each other. It's the homologous pairs that are separating.
So each pole gets a full set of chromosomes, but each chromosome still has 2 chromatids.
Exactly. And because crossing over occurred, those sister chromatids are likely no longer identical. Telophase I follows, where the chromosomes might decondense a bit, and new nuclear envelopes may form around the 2 groups of chromosomes, creating 2 haploid nuclei, technically. Sometimes the cell goes straight into meiosis II, sometimes there's a brief interphase.
Okay, that's meiosis. I separate the homologous pairs. What about meiosis II?
Meiosis II looks much more like a standard mitotic division, but it's happening in those two cells produced by meiosis I, which are already haploid in terms of chromosome number, though each chromosome still has two chromatids.
So like mitosis, but with half the starting chromosomes?
Pretty much. In prophase II, the chromosomes condense again if they relaxed. Metaphase II, the chromosomes, each still with two chromatids, line up individually on the equatorial plane, just like in mitosis.
Centromeres right on the line this time.
Yep. Then, anaphase II, the centromeres finally separate. The sister chromatids are pulled apart, and now they're considered individual daughter chromosomes, moving to opposite poles.
And telophase II finishes it.
Telophase II wraps it up. New nuclear envelopes form around the four sets of daughter chromosomes, the chromosomes decondense, and typically cell walls or membranes form, dividing the cytoplasm. The final result: 4 distinct haploid cells.
4 haploid cells, each genetically different. That's the key outcome, right?
Absolutely. And what makes them so different? 2 main things during meiosis. First, that random orientation of bivalents back in metaphase I. Which way each pair lines up, which homolog goes to which pole, is completely random and independent of how other pairs line up.
How much variation does that create?
A huge amount. If an organism has n pairs of chromosomes, there are 2 to the power of n possible combinations just from this random assortment. For humans, with n23, that's 223, which is over 8 million possible combinations of chromosomes in the gametes.
Over 8 million. Just from random sorting. Wow.
And that's before you even factor in the second source of variation, crossing over. That shuffling of segments between homologous chromosomes creates entirely new combinations of alleles on each chromatid. So the sister chromatids separated in meiosis II aren't even identical anymore.
So random assortment and crossing over.
Together, they ensure that virtually every single gamete produced is genetically unique, different from the parent cell and different from every other gamete. This is the raw material for evolution.
Okay, let's quickly contrast meiosis and mitosis again, just to be crystal clear.
Good idea. Key differences. 1. Divisions. Meiosis has 2 nuclear divisions. Mitosis has 1. DNA replicates only once before both. 2. Chromosome number. Meiosis makes 4 haploid nuclei, half the chromosomes. Mitosis makes 2 diploid nuclei, same number as the parent. 3. Genetic identity. Meiosis products are genetically different. Mitosis products are genetically identical, barring rare mutations.
That difference in genetic identity is huge. Meiosis generates diversity, mitosis generates clones.
Precisely why sexual reproduction is so important for adaptation.
This brings us perfectly to Gregor Mendel, the father of modern genetics. How did he figure out inheritance without knowing any of this cell biology?
It's truly remarkable. He was working in the 1860s. He knew nothing about DNA, chromosomes, or meiosis. But he understood that the characteristics of organisms, like his pea plants, must be determined by inherited factors. We now call those factors genes, and the different versions of those genes alleles. Alleles are found at the same spot, or locus, on homologous chromosomes.
And he used peas.
Yes, garden peas, Pisum sativum. His genius was partly in choosing his organism. Peas have lots of clear-cut, contrasting traits, like purple versus white flowers, round versus wrinkled seeds. And crucially, his method was brilliant. He made large numbers of controlled crosses, and he followed traits through multiple generations, carefully counting the offspring.
Controlled crosses. How?
Peas normally self-pollinate. Mendel painstakingly removed the pollen-producing anthers from flowers before they matured and then manually transferred pollen from a different plant using a small brush. Complete control.
Okay, so what did he find in his first experiments, the monohybrid crosses?
He focused on one trait at a time, say seed color. He crossed pure-breeding yellow-seeded plants with pure-breeding green-seeded plants. This first generation, the F1, were all yellow. The yellow trait seemed to dominate.
So he called yellow dominant and green recessive.
Exactly. Green seemed to disappear. But, and here's the kicker, when he let those F1 yellow-seeded plants self-pollinate, the green trait reappeared in the next generation, the F2, and it reappeared in a consistent ratio, about 3 yellow for every 1 green.
That 3.1 ratio.
Yeah.
That was the clue.
That was the huge clue. It told him traits weren't blending. Let's use modern terms. If Y is the dominant allele for yellow seeds and Y is the recessive allele for green, the pure yellow parent was YY, the pure green was YY. The F1 generation were all heterozygous YY. Their phenotype, observable trait, was yellow because Y is dominant, but their genotype, genetic makeup, was Y.
Okay.
When those Y plants self-pollinated, the allele is segregated during meiosis. Gametes could be Y or Y. Combining these randomly gives F2 genotypes in a ratio of 1YY, 2YY, 1YY. Since both YY and Y look yellow, the phenotypic ratio is 3 yellow, YY plus Y, 1 green, YY.
Ah, that explains the 3.1. And he could test this.
Yes, with the test cross. If you have a yellow pea plant, how do you know if it's YY or YY? Cross it with a known homozygous recessive YY. If all offspring are yellow, the parent must have been YY. If you get a 1.1 ratio of yellow to green offspring, the parent must have been YY.
Clever. You can use a Punnett square grid to visualize that, right?
Absolutely. Punnett squares make predicting these outcomes really straightforward.
So from these simple crosses, he derived big principles.
2 major ones. First, the principle of segregation. It says hereditary factors, genes, exist in pairs. One factor comes from each parent, and these pairs separate or segregate during gamete formation, meiosis, so each gamete gets only one. This explained how traits could disappear and reappear.
And the second principle.
The principle of independent assortment. This applies when looking at 2 or more traits. It states that the alleles for one gene sort into gametes independently of the alleles for another gene, assuming they aren't linked together on the same chromosome, which we'll get to.
How did he show this?
With dihybrid crosses, looking at 2 traits simultaneously. For example, he crossed plants with round yellow seeds, RRYY dominant for both shape and color, with plants having wrinkled, green seeds, Y recessive for both. The F1 generation were all RRYY, showing both dominant phenotypes, round, yellow.
Okay, dominant traits showing. What about the F2?
When the RRY F1 plant self-pollinated, the F2 generation showed 4 different phenotypes in a specific ratio: 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green. That classic 9.3.3.1 ratio.
9.3.3.1. Why is that ratio significant?
Because it shows that the traits for shape, round wrinkled, and color, yellow-green, were inherited independently. You got the original combinations, round yellow, wrinkled green, but also new combinations, round green, wrinkled yellow, in predictable proportions. It wouldn't happen if the traits were always stuck together.
It really is incredible. He figured out the fundamental rules of the genetic lottery without ever seeing the lottery tickets of the chromosomes or genes.
It's a testament to meticulous observation and brilliant deduction. His work laid the entire groundwork for genetics.
But like you hinted, it gets more complex than simple dominance and independent assortment sometimes. What about linkage?
Right. Mendel's second law, independent assortment, works perfectly if genes are on different chromosomes or if they're very far apart on the same chromosome. But what if they're close together on the same chromosome?
They travel together.
They tend to, yes. These are called linked genes. They don't assort independently because they're physically connected on the same piece of DNA. This was first noticed around 1905 by Bateson and colleagues studying sweet peas. They saw F2 ratios that didn't fit the 9.3.3.1 pattern, suggesting linkage between flower color and pollen-shaped genes.
But linkage isn't absolute. Can linked genes ever be separated?
Yes. And the mechanism is that amazing process we already discussed, crossing over during prophase of meiosis. When homologous chromosomes exchange segments, they can swap alleles between linked genes.
Ah, so crossing over can break linkage.
Exactly. And the frequency of crossing over between 2 linked genes depends on how far apart they are. Genes that are farther apart have a greater chance of having a crossover occur between them, so they recombine more often and seem less tightly linked. Genes very close together rarely get separated by a crossover.
Can you use that frequency to map genes?
You bet. By measuring recombination frequencies between different linked genes, scientists can construct genetic maps or linkage maps showing the relative order and approximate distances between genes on a chromosome. Like the map shown for tomato chromosome 1 in the textbook, using map units based on recombination percentages.
Fascinating. Okay, besides linkage, what else complicates the picture? Mutations.
Mutations are fundamental. They are any change in the hereditary material, the DNA sequence, or chromosome structure. Hugo de Vries first coined the term around 1901, observing sudden, heritable changes in evening primroses. He realized this must be the source of new variation.
What kinds of mutations are there?
Lots of types. You have point mutations, which are very small changes, maybe just a single DNA base being swapped, inserted, or deleted. These can happen spontaneously or be induced by mutagens like UV radiation or certain chemicals.
Like typos in the genetic code.
Good analogy. Then there are larger chromosome mutations or structural alterations. These include deletions where a whole segment of a chromosome is lost.
Making the chromosome shorter.
Right. Duplications where a segment gets copied, so it appears twice or more. Inversions where a segment breaks off, flips 180 degrees, and reattaches, reversing the gene order in that section.
Flipping it backwards.
Yep. And translocations where pieces get exchanged between two different non-homologous chromosomes, often reciprocal, meaning they swap bits.
So big rearrangements. What about genes that move?
Ah, yes, mobile genetic elements or transposons, often called jumping genes. These are segments of DNA that can actually cut or copy themselves out of one location in the genome and insert somewhere else.
They actually move around.
They do. Barbara McClintock discovered them in maize back in the 1940s, groundbreaking work that won her a Nobel Prize much later. She figured out that the splotchy color patterns of variegation in corn kernels were caused by these transposons jumping into or out of pigment genes, turning them off or on.
Wow. And that movement itself can cause problems.
It can. When a gene or transposon moves, its new location might disrupt the function of a gene it lands in or alter the regulation of nearby genes. This is called a position effect, and it can definitely lead to phenotypic changes we see as mutations.
Okay, so point mutations, chromosome rearrangements, jumping genes, any other types?
We can also have changes in the number of chromosomes. Aneuploidy is when there's an addition or loss of just one or a few individual chromosomes, like having one extra or one missing. Think Down syndrome in humans, which is trisomy 21.
An extra chromosome 21.
Right. And polyploidy is even more dramatic. It's the duplication of entire sets of chromosomes. So instead of being diploid 2, an organism might become tetraploid 4n or hexaploid 6n. This is actually quite common in plants, often leading to larger cells and sometimes new species.
So why are mutations, which sound like errors, actually so important?
Because they are the ultimate source of all new genetic variation. Without mutation, evolution couldn't happen. Crossing over and independent assortment shuffle existing alleles, but mutation creates new alleles in the first place.
They provide the raw material for natural selection to act on.
Precisely. In a diploid organism, a new mutation on one chromosome might be recessive, or its effect might be masked by the normal allele on the other homolog. This allows the mutation to persist in the population's gene pool, potentially becoming beneficial if environmental conditions change.
So these random changes happening at a low rate, maybe one mutation per 200,000 cell divisions in eukaryotes combined with recombination, provide enough fuel for evolution.
That's the idea. It's a continuous process.
Now let's dive into gene expression. It's not always simple dominant recessive, is it?
Not at all. Sometimes you see incomplete dominance. Here, the heterozygote's phenotype is an intermediate blend between the two homozygous phenotypes.
Like mixing paint. Red and white make pink.
Exactly like the classic snapdragon example. Cross a homozygous red-flowered snapdragon, RR, with a homozygous white one, RR, and the F1 generation are all pink.
Does this mean the alleles blended?
Nope. That's the key. If you self-pollinate those pink R plants, the F2 generation shows a 1 red R, 2 pink R, 1 white R ratio. The red and white alleles segregated cleanly, just like Mendel predicted. They didn't blend. The heterozygote just has a different phenotype.
Okay, so alleles stay discrete. What about having more than two options?
That happens too. While any individual diploid organism only has 2 alleles for a gene, within a population there can be many different versions or multiple alleles of that gene. A good example is self-incompatibility genes in plants like red clover. There can be hundreds of different alleles in the population, preventing self-fertilization and promoting diversity.
Mendel was lucky his peas didn't have that.
He really was. It simplified things considerably for him.
And genes don't work in isolation, do they? They interact.
Absolutely. Most characteristics aren't determined by a single gene, but result from complex interactions between 2 or more genes. One important type of interaction is epistasis.
Epistasis with that.
Epistasis is when one gene masks or interferes with the expression of a completely different gene at a different locus. Think of a Labrador retriever coat color. One gene determines black versus brown pigment, but a second, separate gene determines whether that pigment actually gets deposited in the fur. If the dog is homozygous recessive for the second gene, it will be yellow regardless of what the first gene says about black or brown. The E allele is epistatic to the black-brown alleles.
So one gene can override another. The textbook mentions foxgloves.
Right, a similar idea. In foxgloves, one gene, DD, affects the intensity of red pigment, but another gene, WW, controls where pigment is made. The dominant W allele restricts pigment to small spots, effectively masking the effect of DD on overall flower color. A cross involving both genes gives a modified 9.3.3.1 ratio because of this masking effect.
Okay, epistasis is one gene affecting another. What about one gene affecting multiple traits?
That's called pleiotropy, a single gene having multiple, often seemingly unrelated, effects on the phenotype. Mendel even observed this. He noted that in his peas, the gene controlling flower color, purple versus white, also influenced seed coat color, gray versus white, and the presence of a spot on the leaf axils. One gene, three traits.
And sometimes many genes contribute to one trait.
Yes, that's polygenic inheritance. Traits like height, weight, skin color in humans, or yield and size in plants aren't controlled by just one or two genes. They result from the cumulative, additive effects of many genes, often interacting with the environment.
And these traits show continuous variation.
Exactly. You don't get just two or three distinct categories, like Mendel's peas. You get a whole range, a gradation of phenotypes, often producing a bell-shaped curve if you graph the distribution in a population. Early experiments with wheat kernel color beautifully demonstrated how multiple genes could interact to produce a spectrum of red shades.
So genetics isn't just in the nucleus, right? What about mitochondria and chloroplasts?
Excellent point. Those organelles, plastids like chloroplasts and mitochondria, actually contain their own small circular DNA molecules with their own genes. Inheritance controlled by these genes is called cytoplasmic inheritance or sometimes extranuclear inheritance.
And how does that get passed on?
Because the egg cell typically contributes almost all the cytoplasm, including mitochondria and plastids, to the zygote, while the sperm contributes very little besides its nucleus, these traits are usually inherited solely from the female parent. This is called maternal inheritance.
Any examples in plants?
A classic one is leaf variegation, those patterns of green and white or yellow patches on leaves of plants like coleus or hostas. It's often due to mutations in chloroplast genes, leading to cells with defective or no chloroplasts. These traits are passed down through the egg cell's cytoplasm.
Any practical implications?
A huge one is cytoplasmic male sterility, CMS. This is a maternally inherited trait, often linked to mitochondrial genes, that prevents plants from producing functional pollen, but doesn't affect female fertility.
Why is that useful?
It's incredibly useful for producing hybrid seeds commercially, especially in crops like corn, sorghum, or onions. If the female parent line is male sterile, breeders don't have to manually remove the pollen-producing parts, detassel corn, for instance, to prevent self-pollination and ensure cross-pollination with the desired male parent line. CMS does the job for them.
Saves a lot of labor. Now, we've mentioned it a few times, but how important is the environment in all this?
Critically important. A phenotype, what you actually see, is always the result of an interaction between the organism's genotype and its environment. The genes provide the potential, but the environment determines whether and how that potential is expressed.
Like a plant needing sunlight to be green, even if it has the genes for chlorophyll.
Exactly. Or think of temperature effects. Some primrose varieties have red flowers at room temperature, but white flowers if grown above 30 degrees C. The genes haven't changed, but their expression has.
The water buttercup example was striking too.
Yeah, ranunculus. Genetically identical plants producing broad, flat leaves above water but finely divided, feathery leaves below water, same genes, different environments leading to dramatically different forms because environmental cues affect things like cell expansion rates. Everything from temperature, pH, light, water, nutrients, hormones, even the effects of other genes, it all plays a role. Genotype plus environment equals phenotype.
Okay, we focus heavily on sexual reproduction and its genetics. But plants have another major strategy, asexual reproduction.
Right, also called vegetative reproduction. This is fundamentally different because it doesn't involve meiosis or fertilization. Offspring are produced from a single parent via mitosis only.
Meaning the offspring are.
Genetically identical clones of the parent plant. It's incredibly common in the plant kingdom. Some species reproduce only asexually, though typically their ancestors were sexual.
And plants have tons of ways to do this, right?
Oh, absolutely. Very inventive. You have things like runners or stolon stems that grow along the ground, like in strawberries, rooting and forming new plants at node.
I know those.
Then rhizomes, which are underground stems that spread horizontally. Think irises or many grasses. Each node can sprout a new shoot, which is why rhizomatous weeds can be so persistent. You leave a tiny piece behind, it can regrow.
Ugh, yes. What else?
Specialized storage organs often double as reproductive structures. Corms, like gladiolus, bulbs, like tulips or onions, and tubers, like potatoes. You plant a piece of potato tuber with an eye, which is actually a bud, and you get a whole new plant.
Then there are suckers, sprouts that come up from the roots of plants like cherries, apples, or raspberries. That's how commercial bananas are propagated too, from suckers.
Even leaves can do it. Some plants, like the appropriately named maternity plant kalanchoe, develop tiny, complete plantlets along the edges of their leaves. These drop off and root easily.
That's amazing. And there's one more type, epimixis. Sounds complex.
Apomixis is fascinating. It's essentially seed production without fertilization. The embryo develops directly from a cell within the ovule, bypassing meiosis and syngamy. So, the seeds produced are genetically identical to the mother plant. Dandelions, some citrus, Kentucky bluegrass, they can do this. It's like cloning via seed.
So plants can hedge their bets. Many use both sexual and asexual methods.
Many do, yes, like violets. They might produce seeds sexually but also spread vegetatively. It gives them flexibility.
Okay, so what are the pros and cons? Why choose one method over the other?
Asexual reproduction is great when the environment is stable and the parent plant is well adapted. It allows for rapid, efficient production of many offspring that are just as successful as the parent. No need to find a mate, simple replication.
But the downside?
The big downside is the lack of genetic variation. All offspring are clones. If the environment changes or a new disease sweeps through, the entire population might be wiped out because they all have the same vulnerabilities.
Yeah.
There's no pool of diverse traits to draw from for adaptation. And if a harmful mutation occurs, it gets passed to all clones.
Whereas sexual reproduction?
Sexual reproduction's main advantage is generating that incredible genetic diversity through recombination and random assortment. Populations are highly variable. This increases the chances that at least some individuals will have allele combinations that allow them to survive environmental changes, resist new diseases, or colonize new habitats. It fuels adaptation.
But it's costly, right? Finding mates, making flowers, pollen.
Very costly in terms of energy and resources, and you only pass on half your genes, whereas an asexual parent passes on 100%. So there's definitely a cost.
Yet sexual reproduction is incredibly common, especially in eukaryotes.
It is. Despite the costs, its prevalence strongly suggests a profound long-term evolutionary advantage, likely tied to that ability to generate variation and adapt. But the precise balance of forces maintaining sex is still a really active area of research and debate among biologists. It's not a completely solved puzzle.
What an incredible journey through the world of plant genetics. We've gone from the microscopic dance of chromosomes, that amazing DNA packaging,
Right, through meiosis with its crossing over and random assortment generating diversity.
to Mendel figuring out the basic rules just by counting P's. Segregation, independent assortment.
And then exploring the nuances beyond Mendel linkage, epistasis, pleiotropy, polygenic traits, even cytoplasmic inheritance.
Plus, the crucial role of mutations as the ultimate source of new variation,
Yeah.
and the constant interplay between genes and the environment shaping what we actually see.
Exactly. It paints a picture of heredity not just as simple transmission, but as a dynamic process involving shuffling, interaction, change, and environmental influence. And we even touched on the whole other strategy of asexual reproduction.
It really highlights the complexity and, frankly, the elegance of how life perpetuates itself, especially in plants. Every plant out there is a product of this long genetic history.
It makes you look at them differently, doesn't it? Thinking about the chromosomes pairing, the genes interacting, maybe mutations lurking, all happening inside those cells. The next time you see a variegated leaf or notice how different plants in a population look, you have a better idea of the genetic stories playing out.
So what's the main takeaway for you, our listeners?
Well, grasping these principles, meiosis, Mendelian inheritance, mutation, gene interaction, environmental effects, it's fundamental to understanding biological diversity, evolution, agriculture, conservation. Pretty much all of biology.
Hopefully this deep dive into the Raven chapter gave you a solid foundation and maybe sparked some new questions.
We hope so. Thank you for joining us on this deep dive into plant genetics. We hope you found it insightful.
We certainly do. From all of us at the Deep Dive and the Last Minute Lecture team, thanks for learning with us.